There is a claim that circulates online. It shows up in videos, in comments, in casual conversation about strange animals. It goes like this. Jumping spiders experience time in slow motion. Their visual world runs at a higher frame rate than ours, so a hand moving to swat one looks slow to the spider, the way it looks slow to a fly. It is a good story. It has been repeated enough times that it feels true. It is not true. And the reason it is not true turns out to be stranger than the story it replaces. Before we get to the spider, it helps to sit with what your own visual system is actually doing. When you look at the world, you are not receiving a continuous flow. You are receiving a rapid sequence of samples that your nervous system assembles into what feels like smooth motion. The rate at which that assembly happens has a name. Critical flicker fusion frequency. It is the point at which a flickering light stops looking like pulses and starts looking like a continuous beam. Exceed that threshold, and the strobe disappears. Fall below it, and you see the flicker. For humans, that number typically sits around 50 to 60 cycles per second, depending on light conditions. That is your temporal resolution. That is how fast your world moves. An animal with a higher critical flicker fusion frequency sees more distinct moments per second. To that animal, the world is more granular. Motion that looks smooth to you would look frame by frame to them. A fly, for example, sees the world at roughly 250 frames per second. Your hand, moving at full speed to swat it, registers as a slow and entirely avoidable threat. The fly is processing time faster than you are. That is not magic. That is hardware. So, where do jumping spiders sit on this scale? This is the moment the common story breaks. In 1990, researchers D.L. Clark and G.W. Uetz ran the definitive behavioral study on salticid temporal resolution. They tested a North American jumping spider called Mavia and Clemens and demonstrated that these spiders respond to video images as if the images are real. That behavior implies a critical flicker fusion frequency estimated at around 40 Hz, and possibly lower. 40. Compare that to the typical human range of 50 to 60, and something quiet becomes obvious. On this one raw metric, the jumping spider is not faster than you. It is comparable, possibly slower. A 2026 study published in Nature Ecology and Evolution by Clinton Harlem and colleagues confirmed the pattern across a much larger dataset. It was the largest comparative temporal perception study ever conducted, covering 237 species from jellyfish to vertebrates. Pursuit predators, animals that chase prey in real time and must correct their trajectory mid-hunt, tend to have high temporal resolution. Jumping spiders, which are stalking, lunging predators, freeze when they commit to a jump and cannot adjust once airborne. They do not need to track moving targets in flight. Their temporal resolution is not extraordinary on the clock speed scale, and evolution, the study argued, appears to have reflected that, which raises the harder question. If the spider is not faster, why does everything about the way it watches you feel like it belongs to a different category of animal entirely? Why does the salticid family have researchers who have spent more than 50 years trying to understand what the visual system is doing? Because time perception is not just about how fast you process frames, it is about what you do with them. And what the jumping spider does with its visual information is unlike almost anything else in biology. The jumping spider family, Salticidae, is enormous. Approximately 7,000 described species, 695 genera. Roughly 13% of all spider species on Earth belong to this single family, making it the largest spider family that exists. They live on every continent except Antarctica. You have almost certainly seen one. Small, compact, with the distinctive habit of turning to face you directly when you approach, like a small, alert animal that has decided you are worth paying attention to. They do not build webs to catch prey. They hunt, they stalk. And at the center of that behavior is an 8-eyed visual system, so divided, so specialized, that it operates less like a single camera and more like 4 separate instruments running simultaneously. You have probably seen one notice you. It happens the same way every time. A small movement catches its attention. It turns. The body pivots to face you directly, and then it stops. Legs planted, front eyes locked on. Nothing about it moves. That stillness is a lie. Behind the cornea of each of those front eyes, something is happening that you cannot see, and that the spider is not doing consciously. Michael Land was the researcher who worked out what. He published 2 back-to-back papers in the Journal of Experimental Biology in November 1969, and the framework he built in those papers still defines how the field understands salticid vision more than 50 years later. What Land found was that a jumping spider's eye is not what you think it is. You probably picture an eye as a ball, a sphere with a lens on the front and a retina lining the back. That is what your eyes are. That is what most eyes on Earth are. The principal eyes of a jumping spider are not eyes in that sense. They are tubes. Long, narrow tubes that take up much of the interior of the head, with the corneal lens at the front and the retina buried deep at the back. The eye is not a window in the surface of the animal, it is a telescope drilled through it. And the retina at the back of that telescope is not a circular sheet. It is a narrow, boomerang-shaped strip, only a few photoreceptor cells wide. The field of view that reaches this strip is tiny, just a few degrees across. But within that tiny field, the resolution is the finest of any terrestrial invertebrate on Earth, down to approximately 0.04 degrees of angular resolution. At the distances jumping spiders typically hunt, that resolves detail finer than 1/10 of a millimeter, sharper than the physics of an eye that size should theoretically allow. In 1980, Williams and McIntyre published a paper in Nature explaining why. Inside the retinal matrix is a pit that acts as a diverging lens, extending the effective focal length by roughly 1.5 times beyond what the corneal lens alone provides. A telephoto system, built into a spider's eye, scaled to a body 1 centimeter long. A telephoto lens with a field of view that narrow is essentially a precision sniper scope. To be useful, it needs to point at something. The spider's head does not turn fast enough to sweep that scope across a scene the way you scan a room with your eyes. So the spider does not use its head, it uses the tube. This is what Land actually watched, looking through the back of the eye at a living animal whose cuticle was transparent enough to let him see inside. The retina inside the principal eye does not sit still. It moves. 6 tiny muscles surround each eye tube, and they drag the retinal strip across the back of the eye, sweeping it across a field of roughly 50 degrees, independently of where the eye itself is pointing. The tube rotates. The retina slides behind it. When a jumping spider appears to be sitting perfectly still, staring directly at you, its retinas are almost certainly moving. Scanning your face in strips, one narrow high-resolution slice at a time, building a composite picture you never see it build. That is what is happening inside the stillness. Meanwhile, the 3 other pairs of eyes arranged around the head are doing something entirely different. They are wide-angle motion detectors. They cannot resolve fine detail, but they cover almost the spider's entire surrounding hemisphere, and when one of them registers movement, it triggers a head turn toward the source. That head turn brings the principal eyes to bear. The peripheral eyes catch the movement. The animal pivots. The precision instruments lock on, and the invisible scan begins. In 2012, a team from Osaka City University, led by Takashi Nagata, published a paper in Science that revealed something no one had described in any other animal before. The principal eye of a jumping spider has 4 distinct layers of photoreceptors, stacked one behind the other. The front 2 layers sit in front of the focal plane. They receive slightly defocused images. Nagata's team demonstrated that the spiders use this deliberate blur as a ranging system. The degree of defocus in layer 2, specifically the green channel, tells the spider how far away the target is. The spider does not use binocular comparison, the standard vertebrate depth perception strategy. It uses chromatic aberration, the fact that different wavelengths of light focus at slightly different distances, as an internal depth gauge. The blur is the measurement. It is a mechanism that has no equivalent anywhere in the animal kingdom. So, what is this animal actually experiencing when it watches the world? We cannot fully answer that. We never fully can with another mind, especially one this different. But we can say this much. A jumping spider processing a visual scene is not doing what you do when you glance at something. It is running multiple optical instruments in parallel, actively scanning with mobile retinas, cross-referencing depth from blur, motion from peripheral detectors, and high-resolution detail from telephoto-level, integrating all of it into a behavioral decision that, in the most studied genus, Portia, involves something that looks remarkably like planning. Portia is a genus of jumping spider found across Africa, Asia, and Australia. It is an araneophage, a spider that eats other spiders. And to do this, it performs behaviors that have made it one of the most discussed invertebrates in the cognitive science literature. Portia will approach another spider's web and pluck the silk with its legs, imitating the vibrations of trapped prey or a potential mate. It waits to see how the resident spider responds. It adjusts its vibration pattern based on the reaction. This is deceptive signaling, trial and error communication adapted in real time. More striking than the web plucking is the detouring behavior. In lab studies, a Portia presented with a target prey spider on a separate platform, with only a complex route available to reach it, will sit and scan the scene before moving. Then it will take the correct detour, including portions where the target is out of sight for the entire journey. The spider is navigating toward a remembered goal it cannot currently see. It is holding a representation of the target's location in some form of working memory and using that representation to guide movement across a route that, from the starting position, requires anticipating what will be at the end of a path it has never walked. Researchers describe this as planning, not metaphorically, but as a functional description of what the behavior requires. In 2016, a team from Ronald Hoy's neurobiology lab at Cornell University, where much of the foundational salticid brain research has been done, published a paper in Current Biology showing that jumping spiders can hear airborne sounds, not through ears. They have no ears, through sensory hairs on their legs that detect the minute pressure waves of sound moving through the air. In lab tests with Phidippus audax, one of the most commonly studied North American species, the spiders reacted to tones at frequencies matching the wing beats of parasitoid wasps at distances of up to 3 meters. 3 meters, for an animal typically under a centimeter in length. The response was consistent, freezing, the anti-predator stillness of an animal that has just registered a threat overhead. And in 2022, Daniela Rosler and colleagues at the University of Constance published a paper in the Proceedings of the National Academy of Sciences that has not stopped circulating in biology communities since. During COVID-19 lockdowns, Rosler was observing juvenile jumping spiders of the species Avarcha arcuata at night. The spiderlings, because they are young, have translucent exoskeletons. You can see through them. What Rosler observed during their sleep was the retinal tubes of their principal eyes moving in the characteristic, rapid, irregular patterns of REM sleep in vertebrates, twitching, directly visible through the spider's transparent body. The same sleep state associated with dreaming in mammals and birds, appearing in a spider with the eye movements visible to the naked eye. The paper was named a Cozzarelli Prize finalist in 2022. The question shifted from whether the state existed to what the spider was processing during it, though whether it constitutes dreaming in any subjective sense remains open. Which returns us to the story we started with. The jumping spider does not experience time faster than you. On the raw clock speed metric that the internet story requires, it is slightly slower. But almost every other assumption embedded in that story turns out to be wrong. The spider is not seeing a faster version of your world. It is assembling a world from parts that have no equivalent in your visual system. Layered photoreceptors used as a rangefinder, retinas scanning independently of eye position, wide-field motion detectors handing off to telephoto-level, airborne sound detected through leg hairs, a sleep state with rapid eye movement in an animal separated from you by more than 500 million years of evolutionary distance. When one of them turns its flat face toward you and holds its ground, the retinas beneath those large front eyes are quietly sweeping across you, building a picture at a resolution finer than 1/10 of a millimeter, while the peripheral eyes track any hand that moves, while somewhere in a brain smaller than a grain of rice, a decision is being made about what you are. That is not a slower or faster version of what you do when you look back at it. It is a different architecture for the same job, arrived at independently, running on hardware you do not share. The internet version of this story asked whether the spider sees time in slow motion. The real answer is that the spider does not need to. It is already seeing something you cannot.